before getting started, just see if there's any questions or things you want to go over for the exam tomorrow. Are there any, if you look at the study guide, were there any topics that you wanted to ask about or just review?
When it says what does transformation do under molecular quantum, what is that referring to?
Yeah, so when you take bacteria and you basically get them to take up DNA, that's called transformation. So with the plasmids, when we were talking about making like a cDNA library or something, at one point you just have plasmids, and then if you want to put them into bacteria, you would use transformation. Yeah.
Could you clarify what the TDE does in the model you did gelatin purposes?
Yeah, so the question is about what it does. Okay. So the TDE is a buffer solution, so part of what it does is to maintain a pH. And it also provides a certain amount of salt there that is conducive for running DNA through the gel. So that's your buffer. The agarose provides the matrix. So depending on how concentrated it is, it can be more, the pores can be smaller, which would allow you, the smaller they are, the harder it is for things to move. So it gives you higher resolution for small pieces of DNA. If you have a lower percentage, it allows the bigger pieces of DNA to go farther into the gel.
Okay, and the last question is, when looking at a plate of worms, what stages can you see all stages except for embryonic stages?
Yeah, you can, so you will be able to see, you'll see eggs, unhatched eggs, so you do see certain stages of the embryo. And then you'll see L1s, L2s, L3s, L4s, and adults. So yeah, you'll see all of them on the plate. Unless it's a starved out plate, in that case you would mostly see the dowers or L1s that are, because they don't have to. Yep. Other questions for that part? Yeah.
What is the one about double strand RNA? And is it like the cell just naturally the siRNA?
So what was your question about can you just say it again?
Yeah, how come it wants to have double stranded RNA?
Oh, for RNAi to happen? Okay, so typically in the cell, there's not double-stranded RNA. Usually RNA would be single-stranded. And so when there is double-stranded RNA, it is seen as kind of a foreign substance. And so that triggers the RNAi response then. And I guess the origin for that would be that many viruses have a double-stranded RNA intermediate. So they want to target that. Okay. So for the exam, it is going to be very similar to other exams that we've had. It's just the content will cover lab stuff. And I think the split between the two, biotechnology and the lab project, is about equal. So it's about 50-50 for biotech stuff, but the biotech stuff is only covering what's on the review guide. So we went over some things that were just kind of interesting, but that you don't have to know, like Illumina sequencing. You don't have to know that. Just you do have to be familiar with Sanger sequencing. I think there's a question about the differences between PCR and sequencing, so we could talk about that. Anybody think of some of the differences if you had to compare Sanger sequencing to PCR? We can start with things that are similar between them. Go ahead.
Something similar that we require DNPs.
Yeah, we'll be right these on the board. So we're talking about so PCR and sequencing. So they both use, oh, this is kind of awkward to write across the middle. Both use DNTPs, and then they both use DNA polymerase, which is heat stable, right? Other things that you can think of that are the same between them? what machine do you use to run the sequencing reaction? Thermal cycler. So we use the same machine for both. Anything else they can think of that's similar?
The way that you have it programs.
Yeah, so the program, so you use that 95 to denature, then you cool it so that the primers can bind, and then you raise it back up so that the polymerase can extend. So I'll say programs are similar. Okay, how about differences between, does similar have an I?
Yeah.
Weird, it does. Similar. Similar. So how about differences between the two?
Sequencing those fragments.
So the product for this is a whole bunch of fragments. Product is variable length fragments. Okay, that's one. How about the product for PCR? What are you trying to get? Is it variable length or no? So it should be a single product is, our goal is to have one product. So it is a single fragment, many copies of a single fragment of a particular length. And that's defined by the primers that you put in. Okay, so another difference, yeah, Ken?
Is that they use different number of primers? One of
Yeah.
them use one, another one uses one.
Yes. Which one uses one primer? Yeah, so this uses one primer because we're trying to extend like this, all from one direction, so we only want one primer. This one uses two primers, which are forward primer and reverse primer, because the goal is to get a copy of the top and a copy of the bottom. So you want two.
What about the packet polymerase?
Yeah, so that could be a difference. Although they both use DNA polymerase, PCR uses TAC, which is, yeah. This one is a little, it induces mutations, so it's not as, it doesn't have high fidelity. So usually with sequencing, we use a high fidelity, which means it won't make mistakes, or as many mistakes. Okay. Any others? There's one other thing I can think of that's a big difference. So they'll both use a template, but yeah.
Well, the Sanger sequencing, don't you have to do further processing of all the fragments?
Yeah, so you will have to, with this, you will have to resolve them, so we can say that. Uses gel or actually it's capillary gel electrophoresis to resolve these and detect. So what are we detecting with this? How do we detect something?
You're detecting the fluorescent light on the 2D entities?
Yeah, so that is the other really big difference, is that this uses DNTPs and DDNTPs, and these ones are labeled. Look at that handwriting, horrible. Labeled DDNTPs. This one only uses DNTPs. They don't use DDNTPs in PCR. Okay, so that's a pretty comprehensive list of differences. I'm trying to think of any other, like, let me look at what I have here. Just so I can help review if there was something that we didn't go over again. Okay, we'll look at the lab exam and see. There was some big thing I never mentioned. So, so the exam has like matching sections. where you match up the name of a reagent with what it does. So just kind of making yourself, based on those study guides, a term list of things that, make sure you know definitions for the things that are on that list or on that study guide. There's questions about when you would use a cDNA library and when you would use a genomic library. So we can talk about when would be a time when you would want to use a cDNA library. Yeah, Daphne?
You can use it when you want to code for some protein.
Yes, anytime that you want to make a protein in bacteria, you have to use something without introns. So those would be anything that says something about expression, or purifying a protein out of bacteria, then you're going to use cDNA. How about for genomic DNA? Can you think of why you might want to use a genomic DNA library? Storage, yeah. So it is storage, and usually it's used with sequencing, like when you're sequencing the whole genome of something. So you want to chop it up, you want to make a library, and then sequence each of the pieces of that. So that's usually when genomic libraries are used. Yeah, so there's some basic, there's basic like knowing what a technique is used for. So with all of the stuff that we've done in our lab project, kind of have to know like why did we do that. So for example, if I said a technique that is used for separating genomic DNA from plasmid DNA, what technique did we use to do that? Or what process did we use to get our plasmid DNA purified away from genomic DNA?
Alkaline lysis.
Yeah, we used alkaline lysis or plasmid purification. We did, but that's called alkaline lysis. Another example, what technique did we use to determine the size of our plasmid? Yeah, you can cut it. Yeah, we used gel electrophoresis to figure out how big the plasmid was. So just kind of recognizing what the purpose, that's kind of if you go through your lab notebook and look at your purposes, that can kind of help you know why did we use that technique. Okay, and then there is a question about alkaline lysis and knowing what was happening, the order of those. So I would say definitely go back through that lab, look at sort of the order, what were the steps. We had those different buffers, but we talked on the board, we kind of drew out what was happening. So just review what was happening with each of those steps. Yeah, go ahead.
The PE and PV buffers, those were just to wash away the previous buffers, right?
Yeah, so PE has ethanol, it helps to wash salts away. PD is a binding buffer. It's supposed to optimize the binding of the plasmid. Yeah, so that the washes are washing away impurities or salts. Okay. And then there's a question about for the pipettes. So just make sure you kind of review. We had that pipette worksheet and talked about how, describing how you use a pipette, also knowing which pipette you use. So what's the general rule for selecting a pipette? If you were gonna pipette like 20 microliters, you have two options. You could either use your P200 or you could use your P20. Which one would you want to use? P20. So you always go to the smallest, or the one with the smaller volume that is still within the range, because that's more accurate for small volumes. Okay. And then there's just some basic questions I think that we talked about with RNAi. We talked about what are the ways to induce RNAi and germs. What are the 3 ways that you can get it? Yeah, you can say one way and then we'll do one way.
Injecting.
You can inject, right? So you can inject the RNA directly in. What's another way?
RNAi feeding.
Yeah, so we're feeding the bacteria which express the double-stranded RNA. And what's the third way?
Soaking.
Yeah, just soaking them in a high concentration of double-stranded RNA. There's some problem-solving questions. So, based on, we did a plasmid purification, and then we ran a gel. Luckily, most of you you saw the ladder, and you saw your band. So that meant that you were successful. But what do you think could have gone wrong if you had only seen the ladder, but nothing in the plasmid lane? What are 2 reasons that could happen? Yeah. So contaminated, and then would that mean that you didn't have DNA or it got? Okay. Yeah, so maybe you didn't purify it accurately, that there wasn't plasmid to start with. That could be another reason. Yeah.
Maybe no loading guide.
Okay, yeah, so the loading guy helps to get it in. The gel green should still interact with it, but maybe you didn't get it loaded. You know how sometimes it went like whoop out the well, then it wouldn't have run through the gel. So it could be a loading problem, or it could mean that you didn't have anything, that you didn't correctly purify. What could happen, let's say that you run the gel and there's nothing on the gel. You don't see a ladder, you don't see your plasmid, and you know you loaded the ladder. What could explain that?
Maybe you didn't add gel green.
Yes, that could explain it. Maybe you didn't add gel green, so that you can't even see the DNA that was there. What could be another reason? How about setting up, yeah, that's right. Setting up your apparatus, you set the current the wrong direction, so everything went into the buffer instead of through the gel. So with those types of things, those are just kind of like troubleshooting. There's usually not one right answer. There's lots of possibilities. But maybe like go through your lab notebook and just kind of review what we did. So yeah, I think you'll be ready for that. And then there is a CRISPR question, so just knowing how CRISPR works. So for example, when you use the CRISPR system, you put Cas9 and you put a guide RNA, CRISPR RNA into a cell. And then what does the Cas9 do once you insert that into a cell? Anybody remember from yesterday?
Cas9 will go around to see if there's any RNA that is similar to the one that it is injected in. And so that means it must be a virus, so that's why they'll cleave it.
Yeah, and it actually cuts DNA, so that's the one. So that's how that one is different from RNAi, but it will go find the matching DNA and then it cuts the DNA. So when you get that DNA break, how can the cell fix it? So once you have a double strand break, what does the cell do to fix it, in a cell that maybe you're trying to do CRISPR with? Yeah.
The HR or NHJ.
Yeah, exactly. So NHJ would be usually the default unless you provide it with a template to use. Then hopefully it will use that template to fix it. Okay, so any other questions that you thought of while we were going through these? No? Okay, so yeah, we'll do 1 hour where you take the test and then you can have a chance to improve your score by discussing. And then, yeah, that's the plan for tomorrow. Then the rest of the time tomorrow is going to be spent picking your L4s, so I don't actually think we'll have time to do lecture outside of that. I know that was difficult.
That was something. I'll try to
I don't know how much I got yesterday.
Just more than me.
Maybe we could do all of it though. Spent 65 days
We had
time to 2-3 hours.
All right. So we're going to start now on cell cycle, which is, I think, a lot of review from mitosis, that you learned in in Biology 160. So we're gonna go through the overview, first of all, of just the whole cell cycle. And then we'll specifically talk about mitosis, what are the stages of mitosis. And the last part is about how do you regulate the cell cycle? How do you tell cells when they should go and when they should stop? And what happens if you don't do that well, which is obviously one of the major causes of cancer, is not regulating cell cycle. So for cell division, what is cell division? Cells are going to divide for multiple purposes, right? During embryonic periods, a cell is going from one single cell to trillions of cells in an organism. So for growth purposes, cells will divide. Once you have an adult organism, cells are still dividing, right? So they are involved in cell replacement, wound repair, and another purpose is asexual reproduction. So cell division is the way that some organisms actually divide or reproduce. This image right here, does anyone recognize what that is? It's a scanning electron micrograph, so it's obviously zoomed way in. Nobody knows what it is? It's used to make bread and beer and yeast. This one is budding yeast, so this is Saccharomyces cerevisiae. And you can see the way that yeast reproduce. They can reproduce asexually just by budding off a daughter cell. So here you see the daughter cell, the mother cell. These little circles are bud scars. So that's previous places where that yeast has formed a bud.
Does that mean that they can't reproduce in that same location?
That's actually a good question. I haven't thought about how many times this cell could divide. You know, like can it divide from that bud again? I just don't know. You could ask a yeast biologist whether they can bud off on the same location again. So those are the purposes for cell division. We're going to primarily be talking about two types of cell division, and that is mitosis and meiosis. And mitosis and meiosis specifically are actually describing the division of the nucleus, but usually they're used as a general term to divide different types of cell division. So mitosis and meiosis, these are the two that go in the blanks right there. They also go in the second pair of blanks, so those are division of the chromosomes. After those divisions, you have the process of cytokinesis, which means division of the cytoplasm and the organelles. So I'll just show you a couple of videos of variation of this process in different embryos. Oh, whoops, I have to stop this. Okay, hold on a second. I find this one video. So for Drosophila, skip that. Oh, we're at the end of it. So one of the cool things about this is that these cells are multinuclear cells in a syncytium. So it's like a bag of nuclei at one point during embryogenesis. And all of these, you'll see they actually enter into mitosis in a synchronized way. So they all start dividing at exactly the same time. And you can see they all go into, in this case, the GFP is highlighting where the chromosomes are. So you see they all like condense and then they split apart at the exact same time. This sort of bag of nuclei will eventually form individual cells at a certain point in embryogenesis. So that's Drosophila, that's our little fruit flies. The other one I like to show you, let me find it. The link for this is embryogenesis, which we did for C. elegans. I can't just visit the link. Sorry, I have to do it this way. Okay. And then I have to escape out of it again. Okay. can see, that's one cell to two cells. And then you can see as these divide, they actually migrate around. So in that single cell, right, the cells, as they divide, get smaller and smaller. The cool thing I think I told you about C. elegans is that the fate of every single cell is known. So they know, even as early as the 2-cell stage, which cell is going to give rise to the germline, which cell will give rise to the other tissue. And you can laser ablate one cell at this stage, and that tissue will not arise. So it's like fated, which is not the same as human development. With human development, you can actually suck a cell out at the 8-cell stage, and they will be able to replace that cell. So anyway, this is, if we kind of speed it up a little bit, look later on in embryogenesis, the cells get really, really tiny from that original cell. And then you start to see the formation of the structure, right? It's starting to look more like an organism as these organs are forming. And then you get to the comma stage, where it looks like a comma. And that's the little worm in there moving around, and then it's going to hatch out. So about halfway through this process, this gets laid outside of the mother and finishes developing on the plate. So anyway, this is one of the reasons that it's really useful to study processes in the C. elegans embryo, because look how big that cell is. And then you saw how small those cells got towards the end. Yeah.
I remember when we did the assignment where we watched these videos, there was another video where the cells were dividing simultaneously. Is there like a difference between, I don't know, types or something?
So they should be stereotypic, as that, the pattern of development should be the same. Even the way that they reorient should be the same every single time. So yeah, they should divide in the same pattern in every single embryo.
Okay. So the one that wasn't doing that maybe had.
Oh, yeah, I guess I didn't realize that was it that the timing was off, or?
Well, like one cell was divided and then the next. I
Oh.
don't know, there's two videos and they weren't.
Okay, they weren't exactly the same.
I don't know.
Yeah, I should check back on that other one, yeah. Okay. All right, so for mitosis and meiosis, let's just see if we remember this from Bio 160. So for mitosis, what type of cell undergoes mitosis? So what type of cells are produced by mitosis? Somatic cells. So you can put somatic in that blank. Somatic cells are produced. And then how about for meiosis? This is for mitosis. Yeah, germ cells are going to be produced, or the sac cells are produced by meiosis. So the gametes, we talked about egg cells and sperm cells. The amount of DNA, so when you undergo mitosis, the daughter cells do they have the same amount of DNA, or do they have half the amount of DNA? What'd you say?
For which one?
Oh, sorry, for mitosis.
They have the same.
Yeah, they have the same. So if we're starting with diploid, for mitosis, it's going diploid and it's creating another diploid cell. They have the exact same. In fact, it's not even just diploid to diploid, but it's genetically identical. So even down here, the daughter cells are genetically identical to the parent. So you're essentially getting a clone of the parent. For meiosis, daughter cells have half the amount of DNA as the parent. So they go from meiosis, they go from 2N to 1N. And they are genetically variable. So you could say you're going to produce different daughter cells. In meiosis, the goal is to generate genetic diversity, right? Okay, so now we're going to go through and review the cell cycle. Okay, so we'll talk about these two slides and then we'll work on number 1 on the ICA. So cells have these stages that they go through. And initially, people really knew that there was a period when cells were dividing, and then there was this intermediate period when they weren't dividing. So they called that interphase. All of the stuff outside of division was called interphase. And then they sort of realized that there was more going on with interphase. So there was a gap phase, which is called gap 1, first gap, G1. And then DNA was duplicated, so that's called synthesis phase. And then there was a second gap phase before division. So interphase is comprised of G1, S, and G2. And then mitosis or meiosis has these different phases. So we'll talk, obviously for meiosis, this goes through this process twice, but they share those different stages. So what I wanted to try to do, just to see if this is, if you remember this, is draw a cell with 4 chromosomes in each of these phases. So basically you're gonna draw a cell in G1, in S, G2, and then you can pick a random phase of mitosis, whatever your favorite phase is. And you're trying to represent what do the chromosomes look like? Is the nuclear envelope intact or not intact? And then also try to see if you remember what was happening with the centrosomes. How many centrosomes did they have? So just to start you off, for G1, you have a nuclear envelope, the cell, and then you have 4 chromosomes. At that point, would there be 1 copy or 2 copies of the DNA? There's only 1. So we would have 1, 2, 3, 4. They're single chromatids at that stage. At this stage, would there be 1 centrosome or 2 centrosomes? You have to think at the end of mitosis, how many did each cell get of the centrosomes? There used to be 2 in mitosis. They each get 1. So you can draw your little centrosome in there as well. There's only 1. Okay, so let's take a few minutes and try to draw this cell, but show what happens with the DNA. And if you can remember, like when the centrosomes get duplicated. And feel free to, if you want to check with your neighbors and just, you know, if you aren't so sure about when things are duplicated, you can always talk to each other.
and then it's, I don't know.
If I,
I saw one and then, yeah, in this one I have two.
yeah, I remember.
So that's in G2.
Something like the G2, something like this, cell phone.
Oh, okay.
It is different. That's why I remember asking about it, but she was
I know I have mitosis.
Okay,
There are two
there's
cells.
two cells. Okay, that makes sense.
G1 is one
She
cell.
says one cell.
S is when it duplicates the
It
number of
is,
zones.
yeah. And then
And G2 is
G2
what?
Mm,
It's a different phase because in, okay, so this here is prepping for mitosis. So there's a check mark, there's a checkpoint where the cell checks out in G1 phase. And I remember my teacher saying that after the S phase, everything is a shift because everything has to do. So no one checking until mitosis.
Okay, so let's see what you have. I know some of you are done, some are still working, but. For S phase, what's the difference between your G1 picture and your S phase picture?
Sister chromatins.
Yeah, so since the DNA has replicated, now each of those chromosomes should have a sister chromatid. So now they have a duplicate. Okay, anything else that happened during S phase? There's something else that gets duplicated. Centrosomes, yeah, so those centrosomes there, each of the centrioles gets a new daughter centriole. So the mother centriole has a little tiny daughter centriole that forms. Initially, they're still together, so if you were to look in a microscope at this stage, the centrosome would almost look like there was only one, because they're still so close to each other. But later on, when they separate, we'll see that there's actually two of them. So the duplication of centrosomes happens at the same time that you're duplicating the chromosomes. Okay, for G2, is there anything that changes about your picture? How about for the chromosomes? Anything that changes for the chromosomes? No, so we're not changing the number. The only thing that really should change is that the volume of the cell should increase, because our goal is to create two identical cells. There is an increase in cell volume in G2. And you're kind of duplicating some of your organelles. So you kind of have to accommodate the, all of the stuff that's being created. But the DNA looks the same. I'll do 4 different chromosomes. Your centrosomes are still duplicated. They haven't moved apart yet. Towards the end of G2, the centrosomes are going to start to migrate apart. You're also going to start to see condensation. So that's when the chromosomes are going to start to thicken and actually be visible as threads that you could see on a microscope. So for mitosis, you could pick any stage. So depending on what stage you chose to draw, your picture might look, different. Let's just say we use metaphase right in the middle. Anybody remember what's happening at metaphase in mitosis? Yeah, go ahead.
Chromatins are lined up.
Yeah, so those chromosomes are lined up along the metaphase plate. They're perpendicular, I have to draw it like this. They're perpendicular to the plane of the mitotic spindle. So one pole has one centrosome, the other pole has the other centrosome. The centrosomes are organizing the mitotic spindle, which is microtubules that are attaching to each of the chromosomes. So this would be a representation of metaphase. In this phase, is there any nuclear envelope at that point? No, there's not. So the nuclear envelope would have broken down in prophase, which is the one right before, the very beginning of the mitosis is when the nuclear envelope starts to break down. Okay, so kind of to review what we just said for these different phases, the numbers here, the time scale is based on human cell cycle. So about 7 to 9 hours for the first gap phase. The major events that are happening during this phase, you have basic metabolism of the cells going on. So you have protein synthesis that's going on. You are beginning, if you're committed to dividing, you have to prepare and make those DNA synthesis proteins. They have to be ready to go. So that'll happen in G1. At that phase though, the DNA is very, very, so when we draw it as lines like this, we're kind of almost oversimplifying. We're saying that it's more condensed than it actually is. What you really should picture in GAP1 phase is a bowl of spaghetti. All of the chromosomes are really long and they're sort of jumbled around. This is a chromosome that has been painted. This is called chromosome painting. Each of the different chromosomes has a probe that recognizes it, but you see that it's diffuse. So it's just this really long strand at that point. During S phase is when all of those chromosomes are going to replicate and get sister chromatids, right? So that's when DNA replication happens. The other thing that happens for S phase is centrosome duplication. So that's when mother centrioles are going to build a daughter centriole. And then we get to GAP2, second gap phase. There's volume increase. Organelles replicate and prepare for division. So those are kind of the major things that are happening there. And that would be the shortest of the phases. The one that I added here, G0, remember we said that most of our cells are actually not actively dividing? G0 is what, I like to think of it like the cell phone parking lot at the airport. If you're picking somebody up but you don't want to drive in circles around, you just wait until they call you and then you go get them, right? That's what G0 is. It's saying it's not actively dividing, but it is ready to go. If there are signals that tell it to go back into the cell cycle, it will re-enter the cell cycle. So they need external signals to go back in and start dividing again. But in most of our cells are in G0. Okay, so we did this part. Our picture is even more complicated than their picture because we added the centrosomes here. So here we have 4 unreplicated chromosomes. And then the sister chromatids are shown. And then now, at the start of mitosis, you see they have condensed and they look like threads. And then they would, of course, divide after that. Okay, anybody have any questions on that first part? Okay, so some terminology that we're going to add in here. We've already kind of said these words, but I just want to make sure that you understand what they are. So each copy of the chromosome is called a chromatid. I use the word chromatid to be a single DNA molecule. So I know sometimes people think that you can only have sisters, so it can only be after it's duplicated. But truthfully, as long as you have a single DNA molecule, that is called a chromatid. So you can have one chromatid, or once it's duplicated, they're called sister chromatids. So for this second blank, you would say sister chromatid. So let's go back over here to this picture. In this cell, we said there are 4 chromosomes, right? That was what it said, draw a cell with 4 chromosomes. How many chromatids are shown in this picture? So just count up how many DNA molecules do you see? 4, right? There's 4 total. So 4 chromatids. Now when we go over here, this is showing how many chromosomes and how many chromatids. Yeah, so it's still 4 chromosomes. The definition for a chromosome is a piece of DNA with unique information on it. I haven't actually created a new set of information. I just made a Xerox copy. So this is still 4 chromosomes. But the chromatids talks about how many copies are there. Now there's 8 chromatids. So the only thing that's happening in mitosis is that we're making an extra copy, and then we're dividing those copies into 2 cells. So G2, what would be chromosome and chromatid for G2? Nothing changed, right? Same as this. So 4 chromosomes and 8 chromatids. Right? Then, when we get here, we still have our 8 chromatids. They haven't divided, but after cytokinesis, it's going to be back down to 4 chromatids. So I want to emphasize this because we're going to get into these complicated things, especially when we talk about meiosis. In this first situation, we were looking at a diploid cell. A diploid means that it has 2 sets of chromosomes. So it was 2N. The ploidy number, how many sets, it never changes during mitosis. So that was 2N, this is 2N, this is 2N, this is 2N. The only thing that has changed is the number of chromatids. Does that make sense to everybody? For mitosis. Okay, and in this picture, they really try to represent what is happening with the DNA, showing you that it's really deep condensed. This is like a bowl of spaghetti. After S phase, it's still really deep condensed, but now there's 2 copies. When it condenses, it looks like the X that you think of when you think of a chromosome. So then 2 other definitions that we need for mitosis, centromere and kinetochore. So centromere, I actually want everybody, if you have the handout, I want you to actually add a second label and label the other side. In this picture, there are 2 centromeres. This always throws people off, and then they say, well, look at the picture that you showed what? In this picture, there's actually 2 strands of DNA. The centromere is that center region of the DNA, and both strands have them, right? So when they divide, both of the chromatids have their centromere. So make sure that you make a note. This has 2 centromeres. So for the definition, we could say it's a region of DNA, typically at the center of the chromosome, but they can actually be found on either side as well. And this is the site at which the kinetochore attaches, and then we'll define what a kinetochore is. Okay, for the kinetochore is a complex of proteins. So I'm going to write, this is our kinetochore. This thing right here is our centromere. So the kinetochore is a complex. It actually involves about 100 proteins. And these proteins attach to the microtubules. So they're sort of the interface between the DNA and the microtubules that are going to pull them apart. Does that make sense to everybody? So how many kinetochores? If there's 2 centromeres here, how many kinetochores would there be? 2. There would be 1 on each side.
Can you say that again before?
For which one?
Can you say again what they're for?
Kinetochores, yeah. The purpose of the kinetochore is to provide an interface between the DNA and the microtubule which is gonna attach on both sides. And then they'll be able to pull them apart. Okay. So, other terminology, mostly I like to go over this because it all sounds the same, right? We just said centromere, centriole, centrosome, they all sound very similar. The centrioles are these barrel-shaped microtubule-based structures that make up the centrosome. You can see the centrosomes are a structure that consists of a pair of centrioles, and that is embedded in this pericentriolar matrix. So that's a bunch of proteins. So this does not have a membrane around it, it's just a whole bunch of proteins. The purpose for this centrosome structure is to organize the microtubules. So if we were to draw here, coming out of this would be a bunch of microtubules. And during interphase, those microtubules are doing different things, and later on in mitosis, they're obviously used to attach to the chromosomes. Okay. So a couple of things about the centrioles, they always have this perpendicular, they're orthogonal to each other. There's always one that is older, that's called the mother centriole, and the daughter centriole is built off of that. When they go through G1, those two disengage, so the mother centriole and the daughter centriole become unattached, and that is when you can actually build new daughter centrioles off of both of them. I think I have a slide that shows how this happens. So this would be during metaphase. When you go to G1, you can see that that attachment between the two has fallen apart. And each one of these is now licensed to be able to build a new daughter centriole. And that happens in S phase. And then in early mitosis, they start to move to opposite poles, where they're going to serve as a bipolar spindle. So one question I have for you is, what would happen if you got more than one daughter centriole? What if you actually duplicated this more than one time? So instead of 2, let's say you have 4. What would then be the consequence of that when you got to mitosis?
Wouldn't be anything.
So, well, there would be maybe too many things to attach to, right? You'd have like extra things to attach to. So what would happen to the chromosomes if they're attaching to them?
They would get
Yeah.
scrambled.
They would get, yeah, exactly. You're going to be pulling them in different directions. And so that would be a multipolar spindle. In this case, it's showing 3. You can imagine that the DNA is going to get fragmented, or you would end up with aneuploid cells that have the wrong number of chromosomes because you pulled them in too many directions. The same would be true, well, not the same, but you can imagine there would also be a problem if you only had one centrosome and you didn't duplicate it, because now when you get to mitosis, you only have one. And how do you pull the chromosomes to opposite sides of just one chromosome? So remember the first day of class I showed you that video where there was the mutant and it didn't replicate its chromosome or its centrosomes? So having too few or too many is a problem for cell division. So that's why it's critical that you have one duplication event and only one duplication event every cell cycle. And I kind of shared with you that there's a lot of cancers that have problems with the centrosomes. Okay. Anybody have any questions on any of that? Okay. So this next part, I'm actually going to ask everybody to just kind of look through these phases. I guess I don't have prometaphase on here, but prophase is usually considered early prophase, and then prometaphase would be like late prophase. So I want you to, actually let's just divide it up, that will make it faster. So why don't you two talk about prophase? This group of 3 could do metaphase. I don't know if you want to work by yourself or if you want to join them.
already started
Okay, so you can show us about anaphase. And then U4 can do telophase and then cytokinesis. So just think about what is happening during those events.
What was that?
And you can draw cells for chromosomes on
I
this.
don't know either. Yeah, me too. I suck at this. So, where is it? Trying to find it in here.
It's, okay,
Where's
there's
that
this.
one? Nuclear envelope forms and chromosomes condense. So I guess it's after anaphase.
Telophase or telophase, whatever. It's the last phase
The last
of
phase of,
mitosis.
And then that's when the cell division. Oh,
Or the nuclear division ends. And
yeah.
then the cytokinesis, which is where the cell splits.
Yeah.
So telophase is, you have two
Nuclear.
nuclei
It's like
in the same cell.
it's like kind of like this.
And then it cleaves. And then goes forward, I think. Is that right?
It's like after metaphase.
But it looks like
So the telophase was whenever they get cleaved.
Yeah.
Yeah.
So there are two nucleuses, two nuclei in the
So they
cell.
make a new, yeah, because there wasn't any, so they're making a new nuclear,
Yeah.
nuclear envelope.
So basically at this stage there's one cell with two
Two
nuclei
nuclei.
and two copies of all the organelles. And then cell division is when it cleaves.
Yep.
Okay, I think that makes sense.
Yeah,
I
yeah.
think
So,
so.
okay, that's not too bad. Ugh, okay.
Robert? I don't know who Robert is.
Yeah.
Okay, so let's go through these. I know this is mostly review, that's why I don't wanna spend a ton of time on it, but, for prophase, do you wanna share what's going on in prophase? You can use this picture.
So in prophase, the nuclear envelope is
Oh, are you
situated
trying
and chromosomes
Is this
are
like that?
much more visible.
Yeah, so
What did you
one thing is that the nuclear envelope starts to break down and disappears. It's sort of actually the membranes are reorganized during mitosis. And then the chromosomes start to thicken, so they condense so that they're more visible. There's one other
Need to draw
thing you
the,
can see happening right here.
microtube.
Phase.
Yeah, so the centrosomes will move apart. There's motor proteins that push them apart. They move to opposite poles, and they start to send out the mitotic spindle, which is the microtubules that are ultimately going to be attaching to the kinetochores. Okay, so you can see prometaphase is kind of late. Prophase, they're showing nuclear envelope is breaking down, and that's when you actually start to have microtubules capturing the kinetochores. So metaphase is the next one. So what all did you say about metaphase?
The chromosomes like lies along the center of the cells, and then the centrosome like the spindles attached to the what's that called?
Kinetochores, yeah.
Kinetochore too.
Yep, okay, so you start to have, you have complete attachment, it's called a bipolar attachment when there's tension on both sides. We're gonna talk about the checkpoint that makes sure that it's attached on both, from both poles. And because of that tension that's pulling in both directions, they align along the metaphase plate. So you can see nuclear envelope is completely gone, and you get alignment of the chromosomes along the metaphase plate. Okay, So then for anaphase, do you wanna share, Tara, what happens?
The chromatids are being pulled to opposite sides of the cell.
Yeah, exactly. So now you have shortening of the microtubules, which allows these to separate. They go to opposite poles. And then the last phase of mitosis is telophase. So what did your group say happens? in
It's when the
telophase?
nuclear envelopes reform, and you basically end up with two nuclei
Yep.
in the same cell.
Exactly. So now you have two nuclei. This would be a multinucleated cell, right, for the time being. I want to note that the centrosomes are outside of the nucleus. One thing I often see, I don't know why, I don't know, sometimes people just put their centrosomes inside the new nucleus that they create, but make sure that centrosomes are always outside of the nucleus that is created. And you can see that each nucleus has 4 chromatids, so it has the individual chromatids that have been separated. And then last of all, we have cytokinesis. So do you want to explain what cytokinesis is?
Yeah, so this is the cell division stage, and there's a contractile ring or
Yep.
cleavage cell that's forming, and I think that's made of microfilament.
Yeah,
The
exactly.
centrosomes
And
are
then-
not inside the nucleus, they're outside the nucleus.
Yeah, perfect. So the you
Nuclei.
see this is a cleavage furrow as it starts to shorten. It's actually the same proteins that are involved in muscle contraction, so actin and myosin form a band, and then they pinch off. They to basically divide the cytoplasm into two distinct cells. I have a picture of that cytokinesis event. So for animal cells, they do form this cleavage furrow. And the two proteins that do this are actin and myosin. So let me write those in here. Actin, or those are microfilaments, and myosin, those are the motor proteins that interact with actin and they shorten. The organelles in cytoplasm are randomly divided, which means that about half the mitochondria, half of the peroxisomes, all of those different organelles have to be divided. And then finally, you pinch off into 2 complete cells for animal cells. Plant cells can't really do that. What's the reason why plant cells can't use the same cleavage furrow formation? They have a cell wall. So instead of forming this furrow, they have vesicles that deliver bits of the cell wall components to the middle. They form a cell plate. Basically install a wall between the 2 cells rather than pinching off. So that's sort of the difference between those 2. Okay. So I'm going to just go into a little bit of this and then we'll take our break. And towards the end of, we'll talk about cancer towards the end of class. But so if we look at the cells in our body, we said that a lot of them are not dividing. Can anybody think of a cell type that does not divide? Yeah, neurons, right? So neurons are one cell type that does not. So a lot of our cells are called post-mitotic or non-cycling cells. Those are the ones that are not dividing. We do have cells that are actively dividing at a very quick rate. Anybody recognize what the structure is here? Where that would be found in the body?
In the intestine?
Yeah. Yeah. So those are the villi that you have lining your small intestine. Those cells are dividing at a very rapid rate. So in fact, they say that you turn over your intestinal cells in 5 to 6 days. So you are constantly dividing those cells. You also are creating blood cells at a sort of constant rate, and they say you turn over those in about every 3 months you have a whole new set of blood cells. So those cells are undergoing rapid cell division, and then one type of cell that undergoes even faster is embryonic cells. So this is the C. elegans embryo. They cycle so fast that they don't even go through the gap phases. They just go S phase, duplicate, M phase, divide. and it happens so rapidly because they are, creating, millions or trillions of cells. So how do those three different cell types know when they're supposed to divide? Whether they should divide, when to enter the cell cycle, etc. So when people were trying to figure this out, the earliest experiments, they would take two cells that were in different phases. So they would take a cell that was in mitosis and a cell that was in interphase. And they used chemicals to fuse the cells together. If you take polyethylene glycol and treat cells with that, the membranes will actually fuse. When they did that, whenever they took a mitotic cell and mixed it with an interphase cell, the interphase cell would always start to go through mitosis. Didn't matter what part of interphase it was, it always would start going through mitosis. So they said, all right, well, maybe there's something in the mitotic cell, it's going into the interphase cell, and it's starting it in mitosis. So they wanted to figure out what was that thing that was going from the mitotic cell. So they did a series of experiments where they actually were working in Xenopus. Xenopus is this, I think they're gross. never want to do experiments on this thing. I mean, you have to, that's a big frog. So this is a South African clawed frog that a lot of people do experiments on because, again, the eggs are huge. So biochemically, it's a really good source of a lot of proteins. But what they did is they would basically take a Xenopus egg cell that was at different stages of the cell cycle. And they would take the cytoplasm from one of these cells, because they're so big you can actually suck out the cytoplasm. So they would take the cytoplasm from an M phase cell, mitotic cell, and the cytoplasm from an interphase cell, and they inject that into an interphase cell. Anybody have any predictions based on the cell fusion? What would happen?
It would force it to go into mitosis.
Yeah, so which one? This one or this one?
The
What do you think? If the,
interphase one.
yeah, so let's see what they found when they did this. They did see that. So if you inject cytoplasm from a mitotic cell into an interphase cell, it induced that cell to immediately enter into mitosis. So what does that tell you about whatever it is that is doing this? Where is it found? It's found in the cytoplasm. So it tells us there's some kind of a factor, probably a protein, right, that is in the cytoplasm. So the conclusion from that was that the cytoplasm of a mitotic cell contains a factor, likely a protein, that promotes M phase. They called this MPF, which for them was M phase promoting factor. So that's how MPF got its name. M phase promoting factor. Now they just had to figure out what is this? What kind of a protein is that? It turns out that MPF is in fact a protein. The protein has two parts. So it's two subunits, right? So one subunit is called a cyclin-dependent kinase, or CDK. So the protein kinase part was a CDK. What does a kinase do again? We talked about this in cell signaling.
Like the Oprah of phosphate. Everybody gets phosphate.
Yeah, the Oprah phosphates, yeah. It phosphorylates everything. So it basically puts a phosphate group on other proteins. So that's what that CDK does. And then there is another molecule that's called a cyclin, and that was the regulatory molecule. So the cyclin is required for the activity of the CDK. If you just have CDK, no cyclin, then the CDK doesn't work. It does not add phosphates to anything. So how does MPF regulate cell cycle progression? Well, if you see the word cyclin, hopefully you see the word cycle. The reason that got its name is that they found if they looked throughout the cell cycle, the amount of cyclin changed. So if look throughout G1, S, G2, the cyclin protein levels change throughout the cycle. So they would be increasing until they reach end phase. That would be the peak of the cyclin concentration, where it would be at the highest concentration. And then it would be rapidly degraded, and that would get you back to G1. When they measured the CDK, they found CDK is actually always there. So how does this work is that the cyclin controls the activity of the CDK, and it's the cyclin that gets made in a regulated way. So you only produce the cyclin when it is time to go into M phase. So the conclusion from this, CDK levels stay constant. Cyclins are cycling throughout the cell cycle, and they get targeted for destruction as well. So they get, we haven't talked about ubiquitin, but ubiquitin, I'll write that word, is a tag that gets put onto proteins that causes them to be degraded. So this is like a garbage tag, degradation tag. So the cyclins will get ubiquitinated, and then they get degraded. So what that results in is basically a clock, a cycling clock, and the active CDK is only active during a certain phase. So does this make sense now, why if you added this component to a cell, it automatically would get it to go into M phase then? Okay, so I know there's about 2 minutes left. So how does it, well, let me just talk about how does it do that. If it's a kinase, what do you think it is going to do? What do kinases do? They add phosphates to things, so it's going to phosphorylate different targets. Just to give you some examples for MPF, to get things to go into mitosis, these are some of the things that get phosphorylated. The condensins, that causes the chromosomes to start condensing once you phosphorylate them. You can phosphorylate parts of the nuclear envelope, that gets it to break down. You can phosphorylate the organelles so that they fragment. You can phosphorylate parts of the microtubule proteins, microtubule associated proteins, that helps you to form the spindle. So basically this is like this action signal that starts all the events rolling for mitosis. Okay, and my last slide, I promise, is that there's not just one MPF. They discovered this one that starts things going into mitosis, but it turns out there's a whole bunch of CDKs. CDKs for every cell cycle. For every season, there's a different CDK. So you have CDKs that get you to go into S phase. You have CDKs that get you to go through this transition of G1 to S. And then you have the M CDK. They pair with different cyclins. So the cyclin is always going to be the regulatory molecule, but there's different CDKs for each of the phases, which makes sense, right? They're going to target different proteins. For SCDK, you can imagine you probably have to phosphorylate some things that are involved in DNA replication to get the cell to start synthesizing DNA. So that's the take home here. You don't have to know much about this other than there's multiple CDKs, there's multiple cyclins, and they all target different things. Okay, so we will stop there. So we can take a 10-minute break. And then when we come back, we're going to seed our plates. We'll probably work a little bit on the methods because we're kind of at a point where we've almost done all of the methods for our project. So for the poster, we'll kind of start to make a list of techniques that we've done. And then the very last part of class, we can go through and talk a little bit about cancer and regulation of the cell cycle.